Hi, I'm Herve Ghesquieres. I'm a professor of Haematology at the University of Lyon. So I co-chair the Hodgkin Lymphoma group of Interlymph with James Mackay. So as a physician in hematology, I'm a member of Interlymph when I'm working with the Hodgkin Lymphoma group. My main topic is the role of single nucleotide polymorphism in correlations with the outcome of Hodgkin lymphoma patients. So I participate mainly on the different projects of Ghesquieres. So I share the data from the French lymphoma group called the LISA for susceptibility study, but also I would like to lead Ghesquieres in correlation with the patient's outcome. So in the Hodgkin lymphoma working group, we mainly work on epidemiologic data regarding environmental exposition. We also work on the role of specific virus, for instance, on specific immune conditions. The second topic is the role of inherent germinal polymorphisms, and the role for susceptibility but also outcome. In this context, we perform large analysis with Ghesquieres. And now it is interesting to open new fields with biological studies to better understand the different link between Ghesquieres finding and the pathogenesis of Hodgkin lymphoma. So in all genome of individuals, there are coding gene, and in all the genome there are some variations that explain some difference between, for instance, two individuals. That's why some individuals have brown hair and other individuals have blonde hair. We don't really know what is the exact functions of these variations, because they are in non-coding part of the genomes. The hypothesis is that these variations in the genome could influence the regulations of genes. So the method is to compare individuals without lymphoma to patients with a lymphoma and analyze the role of these genetic variations to know if they were implicated in the susceptibility of a disease. So the Hodgkin lymphoma subgroup of Interlymph produced interesting data on Ghesquieres in 2014. So currently we would like to increase the numbers of patients and the number of controls to have more data and to have more precisions in first results. We well known that Hodgkin lymphoma is a complex disease, and this disease could be stratified by age, by histological subtypes, by the relations with the presence or not of EBV. So we would like to decipher the role of the variations of the genome for all these different subtypes of Hodgkin lymphoma. So of course we need many data to better understand of all these questions.